Pleasure to welcome back Dr. Philipp Altrock, who was formerly here. For those of you who didn't know, he was an assistant professor here and was about to go on to big and better things in Moffitt, and then decided to go back to Germany and do some cool stuff there. And he was telling me today that he's trying his best to establish an IMO type thing over there at Kiel. So that would be fantastic if it worked out. And Philip's still continues to work with a number of faculty at Moffitt, and including in this room. So we're looking forward to hearing a project which we've not heard about before, I believe.
Yeah.
So take it away, Philip.
Yeah, thank you. Yeah, since Heiko now is certainly not coming to Germany for the foreseeable future, the competition is not that steep for good math onco. I was relieved to see he's going to MBS.
Maybe you want to introduce your student.
He was doing that.
Exactly. So there's Samuel. I just briefly talked about it again. Yeah. So Samuel is a final year PhD student in the it's called International Max Planck Research Schools, of which Germany is full of. Since much like Moffitt, Max Planck institutes are not degree institutions, so they can only grant, and universities can grant PhDs. So that we have a joint Max Planck Research School, which is the topic is actually evolutionary biology in a wider sense. But there's a small cancer connection within there, and he's worked closely with pancreatic cancer biologists at our nearby university, the University of Kiel. But this is joined with these two gentlemen. So Jan Müller, who was a postdoc at a lab at ETH Zurich, when he contacted us about this data that I'll briefly share a bit of. And but the former supervisors of Samuel's are not me in this case, but Michael Ratz and Arne Chosen. I do have some conflicts of interest, so I work with people here at Moffitt. I do receive money from a company in San Diego, and I also consult for another CAR T cell company out of Boston. Cambridge. No, Boston. No, whatever. Both. Let me see if I can find this. Right, so topic here, and I don't know, stop me anytime. I hope it won't be as long as last time I was here a year ago, where we somehow got lost a little bit and I was trying to do something on the blackboard. I try not to do this time. CAR T cell therapies, right, for largely hematologic malignancies. In particular, they work very well for lymphomas, because lymphomas have nicely targetable antigens. So the process is about getting T cells either from donors or from patients themselves, and transducing them such that they will express chimeric antigen CAR receptor that will allow them then to uniquely attack cells that express a certain antigen. And this, to me, somehow the biology of what is happening there must be somehow not in contradiction to what we know about infection biology, where T cells expand, they come in different flavors of memory and effector, or even like stem-like states, etc. They're resident to certain tissues. Here they're not. They're injected and they find their way to the tumor. But this top is basically a diagram to remind me and us that somehow we want to implement some basically immunological features. When this is my view of what over time is happening, there's this event of leukapheresis. So when T cells are taken, then ex vivo or outside of the patients, T cells are activated, transduced, and expanded, so that you have enough of the CAR T cells and the desired phenotype, hopefully, and then they're administered. In the meantime, patients are lymphodepleted, so for a variable factors that are beneficial to this one is basically, to put it in very simple terms, to make room for the CAR T cells, which usually won't be there forever. Bless you. Okay. Is this the next slide? Okay, no. And so somehow CAR T cell kinetics in patients, for instance, or in mice, or even in vitro, have been since the dawn of this new type of treatment era that works, at least for some cancers in the clinic, have been of kind of interests for modelists of all kinds, ranging from the pharmacokinetics, pharmacodynamics, to the more evolutionarily biology, ecology inclined people for various reasons. Mainly because if you just focus on the left, right? So what I said is there's this lymphodepletion, which knocks down the overall white blood cell count of a patient, usually then done prior, and day 0 is usually in the clinic, defined as this day when the dose is administered. And usually the dose is actually relatively small, and we rely on the fact that the product, as it's called, the CAR T cells injected, expand in the patient. And so the interesting part about this to me is like it's a living drug, right? It has features that go beyond your monoclonal antibody, your targeted therapy, or your chemotherapy agent. And so they form as is important for any drug development process. They do have an area under the curve, which can be somehow used to characterize the exposure to the drug, which is important to people. But how really to calculate it has been, in the past 3 to 4 years, maybe a main staple of what's happening. But of course, what's really happening is an example network of things that have to do with the CAR T cell population that comes in different phenotypes. As I mentioned, memory T cells, effector T cells, those who largely but not exclusively do the killing. What I'm totally leaving out here is that T cells, as you know, come in flavors of CD8 and CD4, and there are regulatory T cells that are not transduced, but they also impact, well, they maybe do it this way here. So there's just as a placeholder for another, the resident host immune factors, which could be cells, innate or adaptive. They stimulate the inflammation that is driving part of the expansion, et cetera, et cetera. So there's a lot going on. Key thing is, though, you want to know how, for instance, stimulation works, and you want to know how killing works in order to be able to predict as early as possible, given a dose and some other clinical settings around this time 0, how the tumor will respond. Okay.
What about the native population?
What do you mean by that?
T cells.
The T cells.
They play a role.
They do, right? In multiple ways. In some, well, they're in here.
Oh,
Somehow they're part of it. This is more complicated. Okay. I wrote host immune effects because it could be both. It could be inflammatory signaling as well as resident T cells. Yes. I try to make it, yeah.
Some tumor cells, do they edit their genome to remove the neoantigens? Is this like a mechanism?
Exactly. Sure. There's actual evolution going on that is usually, but so all of this happens in a time scale of weeks, often, right? So not much room for that fast of an evolution, phenotypic editing. I don't know, right?
Are there like multiple antigens that are being targeted?
We'll talk about it in this talk. In general, right, for the big clinical successes, there's usually just one, for instance, against any type of B-cell malignancy, lymphomas or leukemias, CD19 will knock out largely, right, all of your resident B cells, be healthy or not, majority of which the malignant ones are in lymph nodes. So again, yeah, slightly different.
can we bring these two in? Let's get you some seats. There's people standing out there. Let's grab some seats.
I'm sorry for you. I mean, I've mentioned is that interestingly, right? There's some interesting reviews. On the left is one that I can definitely recommend. That's written by people at Novartis, who's one of the major distributors of one of these CAR T cell therapies against B cell CD19 positive. So you're ineligible if you're negative for the targeting antigen, right? But B cell malignancies, and yeah, but right, there's a bit of a kind of a modeling zoo. People try to come up with all kinds of explanations. Basically, focusing on this one core thing is like, how can we most accurately describe this curve to come up with these, what originally in drug development people do, is to come up with an area under the curve that somehow characterizes the potency of the drug. Okay.
That one squeezing over there, maybe.
And I love this diagram on the left, or at least the sets of diagrams, right? Because they remind me of what we used to do all the time. Think of each thing here as an ODE, and there's some interactions going on between them, etc., etc.
Good.
To me, I mean, yeah. So what we do is though, right, somewhat simpler, but I mean, it can still be arbitrarily complicated, right? Tumor without treatment just grows. I mean, it's maybe, or the math IMO 1 first day, right? And, but it's still a graduate course. And or then there's, right, now CAR T cells. They also reproduce for some time. So here they don't necessarily reproduce indefinitely, and they have some negative impact on tumor, right? Or this is exactly this. Like there might be like your background other T cells, for instance. Somehow they might interact with your CAR, and there might be still always ongoing weak killing. There might even be a stimulation of that type of killing. Think about some sort of epitope spreading or some stimulation, because now you've knocked down the tumor to certain sizes that reactivate an immune response in your normal lymphocytes. But what's also important to some people is the fact that T cells come in different flavors, meaning some are much better at killing and some are much better at expanding. And both, of course, is important. The expansion is this here, right? And the survival toward the end, and the killing is responsible for the decline in tumor. And that's a lot of what we want to model. But we yeah, the most successes of this types of therapy are, so these are the approved drugs, these are their targets, and co-stimulatory domains. So you do get kind of this artificial T cell receptor, single chain variable fragment here. Then people found out in the history of developing these constructs that you need spacers for more or less successful immune synapse formations, etc., etc. You could hold an entire lecture about the molecular findings about this. It's all basically built on top of the CD3 signaling domain. And a paper I really like that's done some PK actually looks at exactly like this hierarchy, for instance, and how they differently. But you can tell, like if each arrow here is at least one parameter, but you can probably bet that mostly it's 2 or 3 because it's sigmoidal functions. Imagine how many parameters you would want to have here, A, B, how many data points you would have to fit this. When data is generally sparse, these are drugs that are pushed very quickly onto the market with trials in the size of tens rather than hundreds or thousands. So we're developing these models while the field is just building up a patient database, and no one's trying to share anyways, because most of it's held by different companies who don't want to have you look into too deeply into what is going on as long as they don't have to. They have to share it with the FDA, and probably can invoke some Freedom of Information Act types of documents and go with a lawyer to the FDA, and they have to share the entire raw data, which I always wanted to do at least once if I have enough time. Yeah, you can. A very cool paper is by these guys from a biotech company out in Vancouver. That actually looks at the actual responses. So you recognize a very similar pattern here. You have these T cells in different flavors, and they also assume that there's compartments within the T cells. There's memory types of effectors where they differentiate into more and more effective effectors, and then it kills the therapy. And then they're able to map these kinetics to actual responses. So the colors here are the clinical response, which is more or less a vague definition of how their tumors responded after a fixed amount of time. So 180 days, you call it a complete response if the tumor can't be found. Doesn't mean there is no tumor because it's a hematologic malignancy. There could always be something hiding in the bone marrow. But imaging tells you to some degree there's no more tumor. Partial response, if it's within a certain range of less than baseline tumor, but not above a certain threshold, I forgot how much. And then no response, could have still responded, but by day 180, you see higher than 125% of the baseline tumor or something. So they come in this is.
Is that a model?
There is a math model behind this that tries to that has this type of structure. I think it's ODEs, and it has a substructure that is based on in this particular paper based on immunophenotyping these the CAR T cells with single cell sequencing, and then putting them into groups of kinetics that then their actual kinetics and again, this is actually not what is going on at the tumor. It is what's going on in the bloodstream where the traces of the drug or the CAR is available. So there's also yet another process that I didn't mention at all here. This is what is shedding, because what you're actually interested in is at the tumor, where you never know how much they expand. But then they can classify the expansion profiles into subgroups, which then correlate very nicely with the actual clinical response. I think that's the remarkable thing about that paper.
So it's calibrated to the patient data.
In some way, because they have single cell, or they have like in CAR T.
Obviously, there's 9 equations and there's heterogeneity, and is that why the red has such a wide error bar there?
I'm not sure. It might be. I think there's also like maybe there's about 100 patients involved here, and the groups are largely different in subset size, right? So there's definitely.
But they actually fit it to a model, and that was a big part of the paper. That's good.
Yeah. Encourage you to, you should maybe. I mean, I'm sure, and I mean, I met one of the co-authors, not the first author, who's the head of clinical or research and modeling development at this company. I forgot the name. Notch. They're called Notch in Vancouver. Right. But his second author of the paper was in Banff. I remember there was a guy from Vancouver who by now left that company. So he's probably not allowed to talk about it anymore. I'm sure if you want to hear more about this particular papers, and I encourage you, just invite him. He might just show up on Zoom and give a talk about it. He's a nice guy. Well, yeah, I wanted to say something about our paper, blah, 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 which is bad in other things, especially in that regard. So predicting clinical responses, but it was really great, I think, in actually attaching a stochastic extinction process to this entity here, the tumor, which allowed us to use kinetics of this type to actually make a somewhat similar to a trial. This is the gray line here looking progression-free survival curve, and then lets you quantify the probability that you actually achieve something like a cure, and vary certain parameters such as how much the CAR T cell population could expand, or how much you've managed to, if you go back to this diagram, how deep this lymphodepletion goes, or how high this curve here goes, which are definitely also not well understood patient modalities. So that's. So this is in general kind of a shortish 20-minute review on what the field has done in my mind. Some others, go back to YouTube and watch one of Ross Rockney's talks, different modality, glioblastoma in vitro settings, where they also apply this type of therapy. And you'll be able to learn more about it. So we are looking at AML, where this is not as far into clinical translation as for the lymphoma or B-cell malignancies, lymphoma and leukemias with B-cells. And in particular, the project Jan has worked on is about TP53 deficient or mutant AML cells. And this is just a diagram of kind of like where the field now, it's already probably 2 years old, what the field has in terms of challenges. Toxicity is very different, or not necessarily very different, but can be different with the types of CARs for acute myeloid leukemias. And antigen loss is also a different story, because now we're treating a bone marrow disease that largely lives also in the bone marrow. So where maybe some of what Noemi mentioned earlier applies, that there might be ongoing evolution or heterogeneity that can't be measured at time when it's decided to apply this therapy. And some of that is in the variants of TP53 and its deficiency. And there's some early work that show that generally immune response against TP53 variants in leukemia are very different based on its deficiency status. So that is the paper under review that I think once it's published, you can also release this video. So they show that these AML cells harboring TP53 missense mutations are resistant to T cells anyways, and to CAR T cells as well.
Do you have any idea why they are resistant to T cells?
Yeah, you look, I mean, I think they do, and I have to read the paper again. It's in here. So synapse formation is lower, and they also stimulate more of an exhaustion in CAR T cells. So they just manage to stay alive longer, and then CAR T cells tend to become more exhausted. Samuel, do you want to chime in briefly if you have something deeper to share here, or?
There is definitely some something to do with synapse formation and the molecular cascade afterwards, but I don't know it from back of my head.
What is synapse formation?
So in order to kill T cells form an immune synapse to the target cell, the infected cell or the cancer cell, and inject machinery to kill the cell, or somehow inject themselves into the cell, and leading to apoptosis.
That's
So-
facilitated by the neoantigens being expressed.
Exactly, it must be, right? I think a T cell would do it all the time if it weren't signaled to by your normal signals not to. And then any cell with mutations has certain set of antigen that would be presented to your body's T cells, and then of course the original, or I mean, probably this adoptive, right? So all of this is called adoptive immune T cell therapy. That's probably the oldest type of approach for immune therapy, but it didn't take off before, but after the, for instance, programmed cell death ligand immunotherapies that somehow got bigger in the meantime. But historically, I think this is the older part where people have realized, like, if we can just like somehow come up with either vaccines or be able to target something specifically, we can redirect the T cells that way.
Yeah, I mean, I don't know anything about leukemia, but in theory, TP53 mutated cells tolerate misaggregations better, which could allow them to edit their neoantigens.
Yeah, beautiful. Cool.
better.
Mm-hmm. Yeah. So that's the setting, right? So they have they go from an isogenic AML cell line. Is the name in here? Yeah, MOM13, and they are able to have TP53 expressing, or so to say, the mutant here, red deficient cells, and then they let them compete against human-derived T cells or then transduced T cells. And since the market is still open, right? So they try multiple targets. And you could maybe also even combine them. So the target in AML is also not as clear as in B cells. It's not just CD19 or CD22. It's for instance, CD33, 117, 123, or 371. If they mean anything to you, to me they're numbers and letters, more or less. And also I think the pseudo T cell receptors built for these can be of different varieties. So sometimes you'll see one that binds strongly or weakly, and in the data. Okay, and then they expose these cells in vitro to various E to T ratios. So this is one of the major clinical variants you will see a lot in the literature. So so we talk about T cells, right? But here the T is the tumor, and the E is the effector. Where generally the assumption made is that the entirety of the CAR T cell population is somewhat effector cells. So unless there's specific mention of its phenotype, as in this hierarchy of T cells that I mentioned earlier, from now on that is not a subject of investigation anymore. Okay, so basically you have this four 2x2 matrix in a way. You can compare how regular T cells do against the wild type AML cells, against the mutant cells, and then any of the five, because the CD123 comes in two different variants, CAR T cells against either of the two type of AML cells. Okay, and then their paper goes to show how that actually is less efficient, and how it also could be counteracted. So you do get these kind of drug response curves if you look at the killing assay and its efficiency of killing at 1 and 2 days. So again, this is basically Saumil's data matrix. So for each construct that is a target of AML, of which there's 5. Here, this is just 2 differently designed constructs that have been reported in the literature, and they're called weak and strong. I'm not sure exactly what is weak and strong about them, but they basically target the same thing, but maybe the binding affinity is weaker in one or another. That doesn't necessarily mean that they mean the immune synapse is going to be weaker and stronger. Sometimes it's beneficial because many of these bindings are also what Eric Padron sometimes called promiscuous. So they can somehow hop and bind to something else. So promiscuity in terms of the epitope is also a thing. So it might be beneficial if you can jump around a little bit, because then if it's not expressed in abundance, then you have a chance to maybe hop on something similar that is expressed otherwise. Right? I mean, the beauty of, for instance, tumor infiltrating lymphocyte creation is that you don't care about this at all. You just take some epitope by the tumor directly from tumor cells and try to inject it right into the tumor. But in hematologic malignancies, of course, that doesn't work as well or at all. And then each of these come in different initial conditions, basically. That's the E to T ratio. Okay? And then they've been repeated twice and measured at least twice, I think, maybe 3 times in some cases, and measured over the course of 5 time points at day 0, 1, 3, 6, and 10. Because they also came, as I haven't said, but written here, right? They're labeled differently with luciferase and GFP. So you can trace them. And
What are ENT?
sorry?
Sorry?
Yep.
ENT?
At the bottom here, E to T is an effector to target ratio. So this means there's 1 T or CAR T cells for 16 target tumor cells, or 1 and 24, 1 and 30, 1 and 40. But then, due to the expected efficiency of the target, they also, you can tell that they come in different ratios here, right? Often in these assays, for instance, the one that Fred Lock's lab does in this building, probably, they often set out the E to T in a very beneficial ratio. There's often like 8 to 1. So they have massive amounts of T cells per tumor cell just to show that they're really working toward that antigen. They're not trying to be challenging in terms of like how many T cells can knock out one target cell. Yes.
Just out of interest, like based on this data and these cells, like how many targets can a T cell kill in a day? What's the sort of
In a
range?
day?
Yeah.
I know.
Sort of range
I talk to immunologists in industry and from other fields, and people come up with numbers anywhere between 1 and 500. It's hilarious. I'm not sure. I'd love to say 4, but we've with Fred, we've often assumed somehow 4, and back in the day, Greg Kimmel has pulled out the number 4. But I'm not sure.
So my PhD like focused on trying to find that number, so I'm aware of like the crazy values that some people come out with.
Yeah.
I was just wondering sort of like what values are sort of implied by like this particular data set that we have in front of us here.
Tell me, what values of how many T cells, how many target cells can a T cell kill are implied here? I'm not sure. Do we apply anything?
This is not agent-based or individual-based, so I cannot track that number for each cell. I have rates, but I know from a different context. For example, Guhanda and Monica, they believe that this number is somewhere between 8 to 10, like in context of BITE. So like a specific type of antibody which binds to both target and T cells. So when ALL is treated with BITE, one T cell which engages in binding kills roughly around 8 to 10 and then gets exhausted, like on average.
Yeah. So this is he works with a group in Kiel that uses a lot in the lab as well as in the clinic bispecific T cell enhancers. Right, we don't rely on changing the T cells at all, but make it unlikely that your synapse is formed.
Is there any difference in the growth of the tumor when you knock out p53?
Yeah, I think so.
Isn't that a big bias?
Well, it's part of the model.
In terms of resistance, though, because like the way it's displayed is % killing, so it doesn't account for.
Sami, in a null model without you, what is the difference in growth of the knockout versus wild type AML?
Good question. I don't have this information ready in my head. But
well, I think they set it up such that it isn't in this particular cell line with this particular approach. But in reality, it might be a huge difference.
Well, but 6 days is enough time for tumor cells to have a couple of rounds of proliferation. Day one, it doesn't matter, but they're different.
Yeah.
And has this effect been seen in vivo as well?
Yeah, that is in vivo, which we haven't used. But yeah, I should show you a paper in general, maybe because you have a history in leukemia, so you might be interested in it.
I wonder is there any differences in lactate production? Because oftentimes we work with p53 mutant models, and oftentimes you see an increased glycolysis and lower pH can suppress cytotoxic immunity.
Yes, it does. So this is all held entirely constant here with serum oxygen levels and glucose, but then you say that automatically by cell culture changes.
Yeah.
I haven't seen it in their work that they address this, nor maybe, it's a good question. So that would be the next level to measure exactly like sugars and oxygen availability or something in the environment. This is tried to be as constant as possible. The model we're talking about now, and this is where we have ongoing work that is still progressing to some degree, but there's enough results, I think, for us to talk about for the next 10-15 minutes, is an ODE-based approach. So there's no individual-based knowledge about the actual distribution of attack rate per cell across the population. Might be an interesting approach. We've dabbled around this, so basically this builds on, and Joel unfortunately isn't here, but he's co-authored a nice paper about similarities and differences between classical Lotga-Volterra-like ecological dynamics of predator and prey and T cells targeting target cells, or in this case, cancer target cells. One of the big ones is, of course, that T cells don't eat their target to expand, right? So they expand for other reasons. Mm-hmm. They might be stimulated by, but there's many other reasons, right? And there might be even a suppressive element, and they operate at different time scales. So there might be immediate stimulation, but like a week later, it might be anti-stimulatory by what is released. There is no difference in growth between wild type and TP53 knockouts in the experiments. And I think
I think in vitro, maybe this difference doesn't matter because they're just both of them are growing and they're probably at ceiling of the.
And in vivo, it might be.
My guess. Yeah.
And so in winding process, we ended up trying to use Bayesian inference on these longitudinal data to find one specific element of interest of this type of equation. So X is the tumor. All parameters will be fitted, but g, x, y of X and Y is like a function that we will test as basically the variation in our model and do some model selection. You could as well come up with another function on the exhaustion. That'd be interesting, maybe. T cell death here is put into this one constant term. I think here is where you would come up with where if there's a hierarchy of cells from naive to memory to effector, then this would be different. But we wanted to see if in different pairings, right, in this data matrix in a way, across them, what the best function to represent this interaction would be in terms of T cell interaction.
So what's the general goal of the modeling exercise?
What is G? Right?
G that fits this data.
Correct. Of course, there's no such thing as what is G in general. G is G, right? Yeah. But
Well, if
G
Joe
in
was-
light of that data.
If Joe was here,
Only.
G functions would.
Oh, it has nothing to do with that G function.
Different, yes.
No. It doesn't have anything to do with that G function. So that's why it's a small, it's not a capital G. T cell activation, right? Going back to what we know about basic immunology and what people have tried in the literature, right? Could be nothing. Could be just a constant. Could be a local Volterra type here. Then that's also Holling type 1. And I mean, it's a bit embarrassing because Mark has done this 15, 20, 100 years ago.
Are these T cells already activated, or do you mean something else by T cell activation?
Yes and no, right? So somehow they're still heterogeneous in terms of like how actively they will pursue leukemia cells. Yeah. Yes, there are, and again, no, in the sense of the non-CARs, yes, but also we don't really know how much they're activated without having been stimulated by the antigen. Yes.
Are all those CAR T in the model actively engaging with the tumor?
On average, yes. It's an ODE model, so yeah, there might be heterogeneity.
I
on that
was
level.
just wondering if the activation includes like the parts of the population that are not like being able to see the tumor at the time.
Oh, in terms of a spatial heterogeneity, no, we have to assume it's somehow well mixed. In a spatial setting, it will be can be very different. Exactly. Very interesting question that.
And also you say
is
you have
not
no
asked.
estimation of the kill rate of the T cell, but in A, the attack rate, you must have used some value to calibrate A, right?
It's fitted independently, so it's part of the fitting.
Yeah, so it comes out with some sort of idea of how many tumor cells per
Yes,
CAR gets
Yes.
killed.
it does. By inference also. Yes, exactly. Yeah, there's this paper which came up with, I think that's the function we actually used with Greg's work for reasons that I don't entirely remember, but that it had to do with the fact that maybe there's some spatial constraint, and in Greg's model, we're treating lymphomas, so they do have a constant spatial aspect in lymph nodes where most of the disease is, and there might be just simply for spatial reasons competition for how much you can access, right? So there could be this type of dampening that it tapers off with the density of the other immune cells, so that, right? There could also be something like this, like ratio dependence, so just the frequency of both types of cells in the medium or or some that involves both a competitive effect of how you activate and then kill, as well as the typical handling time approach. So you have some attack rate, and those that are attacking, this is, I mean, in this is hidden in this parameter H, where they have some handling time. So while they're attacking, they can't be attacking someone else, right? So that's the time spent of the basically in predator-prey dynamics as well, of the predator with the prey somehow while they're busy, but they're neither dead nor attacking someone else. So that's why handling time is an interesting thing. And okay. to the specifics of the long and windy road of implementing a Bayesian inference here. I have to say I'm a profile likelihood implementer, so I haven't done it myself, but this is the core of Saumil's work here, right? These are the variables we can use for interpretation, the rate of change or tumor abundance, T cell abundance. This is the E, this is the T, I'm sorry. And rate of change of these, rate of change of the others. Background tumor growth rate, we cannot distinguish between birth and death in the tumor, at least not on this level. Basic attack rate, activation rate, T cell death rate, exhaustion rate, all linear, and then depending on what kind of model we have, handling time, how they interfere with each other, and then there will also be standard deviations across the replicates. And Saumil chose log normal priors, informed by some biologically meaningful, basically upper lower bound of possible parameters, and multiplicative error, and then uses basically the package Turing in Julia or through this interface diffbase. And I'm told non-U-turn sampling is an MCM or Monte Carlo approach for how you accept or reject your trajectories in terms of whether they satisfy your likelihood or not, based on what has just happened before as you explore parameter space.
Are you going to elaborate on that?
Saumil, do you want to elaborate on that?
I guess I'm intrigued why it's called Turing.
Why is the package called Turing?
I have no idea why the package is called Turing, but it's a probabilistic programming language framework like Stan, for example. And no U-turn sampling is a special. It's from a special class of Monte Carlo methods called Hamiltonian Monte Carlo methods, and basically there is no accept or reject in there. What you do is you somehow convert this finding posterior distribution problem into a physics problem where there is well and you throw balls in it, and based on the direction and momentum you throw your ball in, it will end somewhere in the ball, and that's your new proposal. And because you do this throwing business, where it ends, it's actually because of this conversion to well. It's like 1-to-1 correspondent to posterior in the well. So there is no wastage, which is why these algorithms are much better than previous class like Gibbs or wherever you have a exception criteria and Metropolis Hastings and things.
Yeah. Exactly. This comes with an initial condition, basically a position in parameter space and an inertia. So you kick it into some direction, and then you collect the goodness of it along its way. And then the no U-turn means like you stop before the U-turn, and then how good it was can be assessed, and then whether you kick it again or not. There's a nice set of lectures by this Max Planck director in Leipzig and an anthropologist / data scientist, McElrath, on YouTube, where he talks about different types of sampling for parameter inference in a Bayesian way. You need a likelihood function, right? So that is clear, right? We use the likelihood function then to come up with AIC and BICs. I mean, the most intriguing thing to me is that you don't get best fit parameters. Okay, we get distributions of parameters, and so and confidence interval has to be rethought in another way. It's like has something to do with your spread. If you're really unlucky, then you get highly multimodal distributions of the posterior, which I don't know if that's too great, but if not, then you can at least confidently talk about somewhat like a confidence interval that covers, say, some percentage of your parameter space, or best fit is replaced by most probable parameter value, right? Or average. It might definitely not be the same. Average is probably not as good. Yeah, and then so we have these different models. We can also, in the terms of here, what he means by Lotka-Volterra activation exhaustion, it's with or without exhaustion term, but they're all linear. But I mentioned the nonlinear types were either a Holling ratio independent. There's Bazooking Harrison, and goes back to the paper with the great Kuznetsov that some of you might know, studying like basic T cell immunology math models from the 90s or so. And some of them do much better, right? Some lower value is good than others. This guy, to my surprise, sucks. So that's the Lotka-Valterra exhaustion, but no activation. Come again?
Not as bad as the second one.
Yeah, no coupling. Okay, so we can rule out no coupling, exactly. So somehow, okay, ratio dependent is interesting because this might hint at the fact that maybe there's something frequency, and then Jeff, if there's frequency dependent dynamics, you might just go replicator, and I don't know, right? So this is super interesting. None of this, right? It's density dependence. But the best fit across models, across conditions and across constructs is this Beddington-DeAngelis. So this one where you do have a handling time that has to do with the fact that you dampen if you have too much prey. You can't arbitrarily kill more, but there's also some competition built in among the target cells, right? And so that's this function. So as an answer to the question, which is the G for this data, that's the G for this data. By as rigorous as this might be or not, at least that's the unique minimum. And this is, for instance, for one of these constructs, we have that for each of the 5. Remember, there was 4 targets plus 1 came in 2 different variants of how you implement the pseudo T cell receptor there. And for each of them, we have these 4 experimental scenarios across multiple different effector to target or T cell to tumor cell ratios. Thus, we conclude T cell activation. Yes, sir.
Just looking at the AIC values on the previous slide, there was one, the Bayesian one, that was really, really high. But then is that not, that's a bad question, sorry.
We can talk about it later. We can talk about it later too. Yes.
Why have 1G across all of the experimental settings? Why not like try to find like rank them or something?
Good idea. Can do too. I mean, there's some ranking in between. We can look at it, but it was, yeah.
Okay.
Here we wanted to go for somewhat like a universal truth. We wanted to go even further. We wanted to go for, right, naturally you would expect that the initial condition shouldn't matter.
Yeah.
But that data didn't come along as nicely. So it turns out that there is still an effect that we don't mechanistically account for.
Okay.
That is, we fit differently a different set of parameter or parameter distributions for each E to T ratio.
I guess I was more asking.
Yeah, no, I know.
But what about is there something? Would we expect the biology to be the same in the different setting, the activation function? No, not necessarily.
Not necessarily.
Okay.
If we though want to only highlight the aspect of biology of TP53 wild type versus mutant, maybe yes.
Okay.
Very good point.
There's a
Somebody, did you hear that?
There's a trade-off.
Yes, I did. And a short answer is that the result that you saw on previous slide, it's like aggregated or accumulated. But what I did was I fitted all of this model case by case. So I have case by case result, which are somehow amalgamated to give an overall result over different CAR constructs. I mean, this is averaged over construct. That's why there is only one result. But I have each treatment, I have different results.
But how is it different?
So for example, some of the constructs, I have some fixed E to T ratio, and in those construct, those E to T ratio, there is no response. It's just flat growing. And in that case, some of the, like for example, Lotka-Volterra activation or exhaustion ones perform better because there is nothing much fancy nonlinear thing happening. But some of the ones there is really like saturation is important, which is why Beddington-DeAngelis or ratio performs the best. But I think the key is that, yeah, I'm adding more parameters, so it should fit better, yes. But AIC and BIC also give some penalty for parameters. Despite those penalties, those saturations in my activation functions are important enough in two or three constructs, which is why the AIC, overall AIC, it comes out this way.
Yeah, so that's been my understanding. So somewhat overall, it is the better one, but there might be the ones that are silly to fit anyways, because somehow there's not much going on in vitro, but we didn't want to exclude any of those longitudinal data sets where you find kind of simpler mechanics. But yeah, we should definitely also come up maybe with a graphical way to show that in some kind of like a heat map or so where the.
I mean, these all of these that are fitting quite well, they look like a fancier version of Michaelis-Menten equation. And if you think about
Holding type 2 is basically.
Sorry?
Holding type 2 is basically.
Yeah.
The question is if it's on which, if it's an X or an Y. What I find super, yes.
I just wanted to ask about the fitting. Can you fit both A and H together, being that they're
Samuel, you want to say that? Renee asked about can you fit A and H in terms of identifiability because they're multiplied handling time? Of course not.
I have a filter based on identifiability, so I have a space of models, and the model only qualifies when all of the parameters are at least locally identifiable for my data. So some of the cases, like for example, I don't remember the name, but there is like X by Y raised to M, and not all of the M, like M raised to 1 or -1, one of them, A and H, one of them comes out not non-identifiable, and that gets removed by my filter. So anything that you see here is past the identifiability test.
Thank you.
Okay. And I mean, maybe in interest of time, we can leave it here. I mean, I think the interesting part to me that at least across these conditions is how relatively bad this type of function does, where you have only competition occur among predators, so to say. That generally, I wouldn't necessarily call this a distinct win of Holling type versus Beddington de Angelis in this, as they're called here, right? They both make sense. And I remember back when Greg and I wrote that one paper on CAR T cells in the clinic, though, and we ended up with this function. Mark mentioned, like, why don't you use that? We didn't. I don't know why, man. Yeah. So people have been wondering, if you go back to classic immunology papers from decades ago almost, like people go back and forth with these type of responses, and some others they slap them on everything, right? So this type of, do we have the ODE again somewhere? Nah. They slap them on here, they slap them on here, they slap them on here and there, and then you end up with these models with like thousands of sigmoidal functions in one X and Y and stuff. I've always found it confusing enough not to pursue it, so I'm happy we did it here. And at least for one of them, in light of this data, absolutely, right? We can say something about what we think is the one that explains at least most of the dynamics. And then we can talk about these effects, like, okay, each dot here is taking samples parameters from the fitted distributions of parameters. A is the attack rate, and runs the ODE model for 10 days, which was the length of the experiment, and then sees how much the tumor has changed, if it's grown or not. And then you can then you could try to read out that maybe how the attack rate, the term in the linear term in the tumor equation, where there's a AX times Y, affects tumor reduction in this model, now that we use this function, okay? Or here, this is how Samuel has decided to plot the comparison between these, how the left violin is always the normal T cells versus the right violin is the parameter value distribution of the CAR T cells in And then, then we had these 4 experimental settings, how they do, and then we would like to pay attention to those cases where they're largely very different, right? So for this experimental condition, for instance, the attack rate with this target here, the attack parameter is much higher in CAR T cells versus normal T cells, etc. So we can try to interpret this data and the biology a little further, and actually maybe go beyond and attach it to what they did then in follow-up experiments in that original paper that I mentioned. That's under review in mice, which we haven't touched the in vivo data at all because it's not as rich and not as usable yet. It's mostly confirmatory of the mechanism of P53 deficiency. Right, so we use this in vitro system that was originally designed to test this heterogeneity question in AML and whether multiple or how and whether multiple CAR T cell targets against AML work, knowing that a TP53 deficiency is generally in favor of the leukemia in immune response, and focused on identifiable subtypes of T cell activation functions. This model popped out as slightly best that has both, but interestingly, mostly, is about competition for prey and some competition for among the predator, if you will. And an interesting finding somehow that we did much better at finding good fits and identifying models when we did a non-pooled fitting approach, which is maybe something for open discussion. In the grand theory of things, you would like to fit, of course, like everything to a pooled way and then and include that into your likelihood function. But because of the heterogeneity of the dynamics across these different initial conditions, that wasn't necessarily possible. So with that, thanks for your attention. Thanks for coming.
Any other questions?
I got a question. So this is Raphael. Hey, so the ratios that come from the data, right? They were very heterogeneous. Some of these look like, there were only a few effector cells per like many tumor cells, and in other cases it was more 1-to-1. I'm just wondering, like, why these ratios were chosen.
Samuel, do you want to answer that? He's smiling such a...
So yes, this has been one of the biggest pain in my life about this subject. Because, like, for example, like, why is C important? Like, will I even see anything to infer C in this data? Like, for example, C is something like interference, right? Like when multiple T cell have only single target cell and they somehow interfere, or like multiple binding, something in that direction, right? But there is no ratio for like 10:1 or 100:1. So can I even begin to infer C from this data? I don't know. But as a theoretician, I would expect that they run like in log space from 1000:1 to 1:1000 or something, right? Because then my parameter inference can actually talk about like this A and H correlation and things like that. But that's not the case, because I think so what they did was like some 4 or 5 set of experiments where they tried all of this different one on a wide range, and then they narrowed the range to one where they saw some response, and that's how they have this random bunch of E to T ratios. And I just have to work with it, because these were not like, these experiments weren't designed in pair to answer some theoretical question or to work with modeling and fitting. They were done for some other purpose, which is why it's like this.
Yeah, thanks.
I have a very basic question, so I apologize in advance. Is the model, the aim, is it to find which of the clone works, like which of the different CAR works best in the context, or is more like underlying factors that are conferring this TP53 resistance in the tumor?
Neither to us.
Okay.
The original design was to figure out more of the, I think, biological mechanics of what makes them evade killing even by CAR T cells and be based on TP53 mutant or not.
So it's
Right?
resistance of the tumor.
That's the experimental model.
Okay.
Yeah.
Yeah.
But it's not evolved resistance. It's like ad hoc heterogeneity constructed this way. Never in mixture. Right? The next phase would be why don't we throw them in the mixture? It's either all this type of AML cell or the other. So there's no competition on that level either. For us, we piggybacked on that approach with these caveats. For instance, the fact that they, of course, right, they wanted to see effects rather than establish mechanisms to go to at least one of these core questions that in none of the more historic recent 4-year modeling of CAR T cells for actually different malignancies too that I talked about in the first 20 minutes, do people ever really think about that type of question? They're really interested in some of it, but not really in a model selection way. And so we wanted to at least go tag one of these factors. Say, let's ask what the literature has provided in different functional forms for that type of activation in this case. Could have also gone for killing. We did maybe, but here we found most bang for our buck. And wanted to rank for these experiments the goodness of fit in a sense from the Bayesian inference of identifying parameter ranges that are reasonable for different such functions.
But ultimately, you're comparing the G function in the wild type versus P53 knockout.
Okay, also, yeah, true, but more both exactly, or in normal T cells or in CAR T cells. Yeah, exactly. Ultimately, for interpretation beyond just okay, now if we found this function, or for any of the functions we tried to fit, we are indeed going back to this 2x2 matrix where we do have either T cells or CAR T cells, and we have a type of AML cell and a wild type. So that you're right. That's kind of.
And when you look at the model between the P53 knockout and the wild type, is G the only thing that is different, or is all the other parameters also changed?
No, they're all different.
Okay.
Yeah.
Thank you.
Yeah. Everything is always all different, but not too different sometimes, right? And then you focus on, and this is why this is all still work in progress, to milk it to its kind of basically massage it to the final degree. Now that this type of model fitting is done, the discussion basically in the conclusion part of this is one of some of its chapters, as well as in the paper we're writing, will be exactly about that. What is the observed differences? Are they stark differences or not, right? And because you can go here, and right, this is kind of the posteriors of the parameters. Left is always the ones for the non-CAR T cells versus so here this is CAR T. No, right? Left is non-CAR, right is CAR.
Yeah, so there is no TP53 knockout on this slide. Left half is always untransduced T cells.
Exactly right, yeah.
Yes, and right half is always CAR T cell.
And this is equivalence in lockout, yeah.
And if you see like this CD123 weak panel, that's what I want. Like that is one of my most interesting thing I stumbled across is basically why do you see this performance bump at that specific E to T ratio and for CAR T cells? Like, is this something coming from population dynamics of CAR T cells and tumor cells? Can we use this as in if we manage to maintain this E to T ratio for this CAR T construct, will we gain like this 10 times more performance consistently? Is this rooted in population dynamics or some not? That is basically one of the interesting questions.
Yeah. So the most translational thought here would be, if you know some beginning heterogeneity, if you could infer it before you treat, would that mean you change your E to T ratio clinically? Because, I mean, the most elaborate thing of all of this is hospital care. You can always make more product. You can't necessarily give it arbitrarily many times. And so you want to get it right once, and it doesn't live forever in the patients. It lives for weeks to months, and then usually it's gone.
How do they kind of assess this or estimate this E to T ratio in the clinic?
So in the drug design, in those pharmacometrician departments at, say, Novartis or wherever, I don't know how they went from, this must have been in a phase like this sometime, right? 10 years ago. And then the trial went down like 5 years ago or 6. When it was deemed somewhat safe. They must have had an idea of what the AUC is based on your E to T. But there's so many other modalities that affect the AUC, kind of the area under the curve of the expected expansion from, say, murine models, that I'm not entirely sure.
There is
They
a
came
way
up-
to calculate from mouse to human how many CAR, like there's a
But it's like the environment, as Sandy said, like the tumor microenvironment, or what Andre mentioned, right? The tumor microenvironment that also leads to more exhaustion, less expansion.
I'm gonna chime in. When you move to mouse, you don't do E to T anymore because you don't have an estimate of the target anymore.
Sure.
You just dose an absolute amount of CAR T. And then from that absolute amount that is proven like therapeutic in the mouse, you escalate that to a human. And then of course you have to have like some sort of therapeutic window. You don't start at the maximum dose. You usually, the first trial is about 5 patients and like they are staggered. So you start with a low dose and you escalate until you see like that the patient cannot tolerate it anymore. But generally my understanding is that you basically you push towards what is considered a maximum tolerated dose for the patient.
So I think my question is not coming across. I'm not asking how would you select E to T to treat? Like, maybe I'm reading it wrong, but it seems to imply like a patient has a bigger tumor and you want to maintain the same E to T.
No, but
then
it's
you
like
need
not
to-
how this is funny because drugs are not given based on the size of your tumor
Yes.
traditionally. Drugs are
Yeah.
given based on your body surface ratio, right?
Yeah,
Or
so
it's
I was
like,
really curious.
right?
So this E to
this
T
is
doesn't
trying
doesn't affect dosing?
It should, but they don't do that. They're trying to do
Okay.
that, dose differently,
Yeah,
or
yeah.
come up with strategies. For instance, with the other types of CARs that are coming from healthy donors, you have a much better chance of making a lot and giving again and again.
Yeah.
And there you really want it, right? I mean, there's so many other modalities in some of the cancers that are treated. I don't know about leukemias, but in lymphomas, you do have lymph nodes that host most of the disease.
Yeah.
And lymph nodes are here. So you get all kinds of responses already based on where your original masses are that show up on the CT. And so this type of heterogeneity is not necessarily taken into account into the dosing strategy, whereas it should. So then you infer an E to T ratio because you know that people have done that. In the Zuma-1 trial, where Moffitt was one of the key centers for one of these anti-CD19 drugs. Bye, thank you. Please saying goodbye. There was a max. There was simply a max based on body weight.
Yeah.
Right? You get higher and higher for very light patients, and then you hit a ceiling, and then this is all the T cells you're getting. Period. No matter how big your tumor is, and no matter if you're much heavier than that. Right. This is typically in allometric scaling for choosing drug doses is done anyways, right? Because you know that there's a difference between actual body weight and ideal body weight. Ideal body weight is something you calculate out of height and weight rather than, or a combination of which, because obese people have not necessarily they're like 2 times as heavy, but their liver or kidney is not 2 times as efficient, or rather the opposite. So right. So drug clearance, which is important for AUC of drugs. And that's again going back to the original thing why people are super hyped about modeling CARs in PK/PD or DMPK or whatever industrial settings because of this, right? How do you explain this curve with the interactions of the drug are so different with itself and with your entire system than many or if not all the drugs that you know. And you have to go back to actual inflammation kinetics to make sense of it, I think, right? And this is also where then the toxicities come along, right? People get hyperinflamed, they get cytokine release syndromes, which in principle is great for the drug, but it's not great for a patient, right? Because it'll expand, and then thus maybe attack, but then you die of cytokine release syndrome. And so there's a sweet spot.
Okay.
And that also is often independent of EDT. So EDT generally is super bad, the inferred EDT. So now we've given them some number times 10 to the 6 CAR T cells. And it turns out they have a leader of tumor, versus this other person has generally, smaller tumor is better response. But better EDT is not always such a great predictor from inference after, interestingly.
Sure.
Because some tumors, even a small one, might be nastier in its microenvironment, and it doesn't, CAR Ts don't work as well on it. So, so this can be controlled here, but not in patients as much yet.
Oh, cool.
Right, yeah.
Thank you.
Super interesting. I don't know, you should all check out if there's opportunities, especially here, come on, right? At some point, they'll just make the CAR T cells across the street for everyone in some back alley little lab, hopefully, right? Rather than having to ship frozen stuff over once to California, ship it, freeze it again, thaw it, ideally, it's made for you overnight. Expand it, and then they can look at your tumor and maybe like
Overnight, I wouldn't trust that.
Just kidding, but but maybe without even having to necessarily freeze and thaw it, if possible. People there's definitely German clinics that have wet dreams about making cars on site, and might or might not work.
Like separate car for each cycle?
I'm not sure. I'm largely thinking, I'm thinking allo, anyways. Yeah,
Okay.
not auto. We can talk about it.
Can I have a quick question? If you show the fits again, because I'm honestly not convinced. Back one.
These fits are
Forward
dummy
one.
picture. Sorry? Huh?
Forward one.
Oh,
It's the
sorry.
numbers, yeah.
Oh,
So, to me, the obvious one is actually to use the ratio, because it's like 2 parameters less, and
ratio is
you're gaining
very attractive.
120 or something, and I don't know what that represents, that score, like in terms of when you look at your fits, right?
Mm-hmm.
But presumably it's not that much.
Great observation, and
And
we are exactly.
Like if I look at that function, it's essentially T cell activation. That's what all you're looking at is B scaled by those two populations, right?
Yeah. It's very tricky if you think about the deeper consequences of the fact that this is ratio to me, but it's super interesting. And exactly, this is what, and it fits great.
So
Yes.
it's telling you something.
Yes, it does, exactly. Yeah,
Okay.
great observation.
Right, well, if there's no more discussion, Philip's going to be here all day. I'm sure he'll be happy to hear from you. I think he's got a fairly packed schedule, though.
No,
No.
meeting.
Okay. Maybe he doesn't, so he's happy to chat. Anyway.
When someone said, like, should we set up a schedule? I was thinking about like the regular visitor schedule when someone gets a handout and they're like in one.
Well, I mean, if you wanted to meet people, I could have arranged those things to be done by the night. So, it's just
I
easy
know.
life, that's all.
He doesn't want to meet with people, so in that case.
Yes.
Anyway, thanks again, Philip.
I'm here. What do I have? Like, I don't